Hello, my name is Jorge Santiago Blay. I'm a research associate in the Department of Paleobiology, and I'm here to talk about Dyers Rule or Dyers Law, and this is a collaboration with the Smithsonian Institution Archives, and the point of all of this is to actually transcribe these little books, of which there are 4 boxes, 40 of the so-called blue books, which most of them are actually not blue. They are very, very frail, late 19th century, early 20th century, and these are the actual annotations of Harrison Gray Dyer Jr., who was a very, very famous late 19th century, early 20th century American entomologist. So, first of all, I would like to try to explain how is that I got into this project. So, being in a Department of Paleobiology, I happened to study fossils of all kinds, fossils of insects, fossils of plants, etc. And someone brought to my attention this, and I have here a photograph of a fossil about maybe 3/8 of an inch long, and about this big, and we wondered what was that, and all sorts of wild guesses were suggested that we don't have time to explain. And finally, one of the many volunteers who worked with me brought from the field, from the northeastern part of Pennsylvania, here in the USA, from the Pocono Mountains, a shriveled larva of a Hellgrammite. And I told her, Judy, that's it. I am pretty sure the head of that thing is very much in the neighborhood of this fossil. But we were sad because all we got was the front end of the animal, the head. And in a very, very childish way, I moaned, saying, I wonder how large the whole larva was. At that moment, I thought, I have an idea. There is something called Dyers Rule that says that there is a relationship, a mathematical relationship between bodily parts that are hard, not only at a certain point in time, but through the life of the animal, of the insect. For example, here we have grasshoppers. Please do not pay attention to the fact that they are different colors and that they are plastic, toys, etc. And here we have caterpillars. Dyer worked mostly with moth and butterfly larvae. He also worked, by the way, with mosquitoes and sawflies. But just to show this very dramatically, let's use grasshoppers. So let's say that here we have the juvenile stages. So when they molt, when they change, they will grow, let's say, by a fraction or by a ratio of 1.2. That is from, let's call this 100. Let's say the next one will be 120. On the next molt, it will be 120 times the same ratio. That is the key thing to remember, 1.2. And those ratios apply to the hardened bodily parts, not to the softer one like on larvae, etc., etc. So it's like a photocopier, a photo enlargement at the same setting, 1.2, 1.2, 1.2, etc., etc. And so we asked the question, how could we test whether this rule applies also to softer bodily parts, in other words, beyond just the hardened parts? Here at the National Museum of Natural History, we have the great fortune of having colleagues who have access to not only knowledge, but a huge amount of specimens. So it's really a blessing to work here. So I approached Dr. Oliver Flint, who is the curator of caddisflies and aquatic insects, and I told him, Oli, I have this question, I have this a proposition. I know it makes no sense, right? Because when we eat a lot, we become wider. So we would expect a very, very weak relationship for the whole larva. But we said, let's give it a shot, and Ollie was a good sport, and he let me have several of these vials full of Hellgrammites, which are aquatic larvae. And here I have one. I'm going to show it to you. Yes, if we collect them in the field and they feel bothered by us, they will bother us by biting. These things can give forceful bites. And so I asked two other volunteers to take measurements, and about 300 of the specimens were measured: head width, head length, and total larval length. A few weeks later, as we were following this, I began plotting the data. I was just floored. A tremendous relationship. And so I began to wonder, in other words, that we could estimate the size of the larva of this thing because we had the width. So the question was answered, and I began wondering, how did Dyer come up with the idea that there were those relationships? Because he, according to the literature, didn't use that many examples. So I began wondering, what's the origin of scientific ideas in this specific case of Dyers Rule or Dyers Law? And so I was directed to the Smithsonian Institution archives, where they told me, Jorge, don't you know that there are tons and tons and tons of books of the so-called field notes, of the blue books of Dyer, and here we have 4 of those boxes full with these little notebooks. And of course the big problem is, and this one example that I am holding is one of the better, with reasonable handwriting that most of us can't read. But once you begin getting to this size, or to pages where he's writing horizontally and then vertically, etc., it becomes a nightmare. So we are doing that to try to, encourage, volunteers to help us translate this and give us insight. Not only, of course, the numbers are easy to read, but give us insight of, not say, for example, what is the name of the moth and the butterfly that Dyer was actually studying. Sometimes it's hard to read that. What other observations was he making? What other thoughts unrelated to the Dyers Rule was he actually jotting down there that would give us some insight of what was going on in his head that made him come up with this idea? In other words, how can we enrich the answer to the question, how is that people come to scientific insights? From my part, what one of the students working with me and I are doing is entering the data into an Excel spreadsheet, because we want to double check that Dyer's data really fits Dyer's rule. Hopefully it does, but and for other insects where this has been actually studied, it fits generally well. But we want to know his data. How does it fit? Another piece of valuable information we want to know is the code. The USNM, United States National Museum, as this museum used to be known in the past, sometimes they, Dyer and many other scientists, had a code, a numerical code, let's say a specimen 3259. And believe it or not, let me take my gloves off. Many times we actually have, and this is an actual drawer with entomology specimens that Dr. Alma Solis has very, very kindly allowed me to borrow for just this presentation. These are Harrison Dyer's specimens. These are over 100 years old, and some of them have actually a code. So all we have to do is match this number with the actual name of the specimen on a book, on a binder that Dr. Solis has, and photograph this. So we would have not only the data, but the actual, sometimes the larvae, the pupa, the casing in which the pupation happened, and the adult male and females. Not only that, we may have a slide prepared by Harrison Dyer himself with the genitalia from some of the specimens. These are extremely valuable for a species identification. It's perfectly standard protocol to have part of the specimen on a slide. Any questions?
So how did you first identify Dyers Law as something that you wanted to study?
I think it was when I began wondering, how did Dyer come up with this rule? For quite a while, I have been fascinated with history, specifically history of science. History was by far my best subject. The social sciences and the humanities were. I'm absolutely passionate about that. And in the last 4 or 5 years, I have been involved in quite a number of history of science projects. I'm actually involved in quite a number of them right now, one of which is Harrison Dyers' rule or Dyers Law. I think what motivated me the most was, just as I said earlier, what went through his head, through Dyer's head, to make him think that there was a relationship. We know that by the end of the 19th century, the application of math into biology was something that biologists were beginning to do. It was kind of, on the air. But maybe the blue books will provide some illumination as to why he did what he did. Any more questions?
Have you found anything unexpected or surprising in Dyer's transcribed blue books?
Yes, I want to mention 2 things. The first one is that the earlier literature pertaining to Dyer's Law or Dyer's Rule says that this was based on a handful of cases. Specifically, I want to say somewhere between high 20s and low 30s, and I thought, wow, that's not really a lot of data. And yet, when I began going through the pages of the field notes, I have estimated, and we have those estimates here, or it had more than what he has been credited to have. And so that was the first, surprise number 1. Surprise number 2 was that in garnering his data, not always his data kind of fits nicely his own rule. Later on, some 20 or 30 years later, began calling it Dyer's Rule. But yes, those have been the two scientific observations that have surprised us the most. More data than what has been reported, and some of his data not fitting that well. Some of it, at least.
So what are some things that transcribers should be on the lookout for in Dyer's notes when they're working on them in the Smithsonian Transcription Center?
Yeah, I think that the two most important will be the name of the species, the scientific name, the binomen. That would be number 1, because we want to associate those observations with a name. And it would be ideal if there is a code to record that, because then not only will we have an association to the species, but to the actual specimen. And we may be able to locate, as I mentioned earlier, the specimens in the collection with the help of Dr. Solis and other colleagues who study moths and butterflies, and the precious genitalia slide, which is essential, so that we can actually eventually share with the broad public what was actually Dyer observing, what were some of his specimens, and do this in a beautiful way with color images, etc. That's our hope.
So one last question before we wrap up. Why is it important to have this information transcribed? How is that helpful for you in your research?
Yeah, because otherwise it's not accessible. It's tremendously difficult to read some of those notes. It's only when people have a lot of experience. I know very few people who can read his handwriting, and I am sure that with a lot of time, which is precisely what I don't have, I could try to transcribe that. But it would be better if I can do the science, and then other colleagues and friends can have that information available to me to put in a scientific paper and then share with everyone else. Thank you. Any more questions? Okay. Thank you. Bye-bye.